Just give me one. Okay. It's a great pleasure to have Franklin Jacoby with us as part of this Philosophical Issues in Space Science seminar series. Franklin works in philosophy of science and is interested in scientific knowledge as a historically situated form of inquiry. Also does work on origin of life and astrobiology and issues in space science, which we're going to hear about today. So without further ado, I'm going to pass over to Franklin, who's going to talk to us today about exploratory modeling and the search for life. So thanks, Franklin. Go ahead.
Thank you very much, Peter. It's a real pleasure to be here today. Thanks for making this possible. I'm a general philosopher of science, and I've come relatively recently to astrobiology, so I'll look forward in particular to your questions and comments in the discussion. I think I'll find that very stimulating, and hopefully you find this a bit stimulating as well. I'm interested in, for this talk, in modeling, unsurprisingly, and in how that can help us shed light on how to conduct astrobiology work, and also how to better study the origin of life. And so that's sort of the general framework here. There are two things I would like to convince you of today. The first is that some exploratory models are epistemically valuable because we do not know whether they have a target. I'll say more about what precisely that means in a moment. And then the second thing I'd like to convince you of is that those models are valuable because they allow scientists to constrain uncertainty. And this is, I think, quite different from some of the things some people have said about these models, and I'll explain how. The outline for today is I'll just provide a little bit of background for the origin of life studies for those of us who are new to the field, and maybe it'll also help show how I'm thinking about this particular field of study. And then there's a particular model which is the heart of the case study, I suppose, which is the GARD model, that is particularly interesting to me. And I'm going to tell you a little bit about how the model works, and I'm going to claim that it is we don't know whether it has a target, and that's what I will call indeterminate. It's an indeterminate model, and I think that's at the heart of why it's a valuable model, and I'll explain why. And part of that value has to do with astrobiology. Even though the model was originally developed for origin of life studies, I think it's been fruitful for thinking about missions looking for life elsewhere in the solar system. And I'll talk about how that's the case. And then I'll situate the views I'm defending in some of the broader literature on modeling. And I think there are two main other ways of thinking about how this model might be valuable. One is analogical reasoning, and the other is modal reasoning. And then I'll wrap up. Okay, so the origin of life is perhaps unsurprisingly a very difficult thing to study for a number of reasons, most of which are probably pretty obvious. We only have one example of life so far, the life on Earth as it has developed. And although we are acquainted with life on Earth very intimately, the origin of life, the origin of living things emerging from physical systems, non-living systems, is very far in the past. And we don't know very much about what it was like so long ago. We don't know what the conditions, with much certainty, what the conditions on Earth were like. We don't know what the physical, non-living molecules or systems were that gave rise to life. And there are a number of candidates. And we've not been able to use laboratory research or other kinds of artificial environments to simulate the emergence of life from physical systems. So it's very difficult. We don't know. There's a lot we don't know, and there's also a lot we don't know about how best to approach the issue, how to study the issue. And we haven't found other examples from other places in our solar system or the galaxy yet. So we are limited. Nevertheless, there are things we can do to still to better understand the origin of life. One is we can continue to look for evidence elsewhere in the galaxy. We might find life, but we also might find precursors to life, things that are almost living or might be living, and that might tell us in turn more about how we emerged on Earth. And I'll say more about this later too. And then the other approach, and these aren't logically exhaustive, of course, there are other things one can do, but these are two particularly interesting lines of research. And the other thing we can do is we can take a contemporary function that seems very important for living things today, and we can try to establish what sorts of molecules or non-living systems could perform that function and later give rise to life. And I'm going to talk a bit more about this second line of research at the moment. So three functions that are very important for living things today are metabolism, replication, and compartmentalization. These aren't necessarily necessary and sufficient conditions for a living thing, but they might be, and they're certainly crucial to life as we now know it. So metabolism we might think of as just loosely processing energy or taking in energy and doing work with it, something like that, to maintain homeostasis or to reproduce or something else. Replication, all life today replicates in some way or another. We've got replication across generations, and then we've also got replications within organisms. So an organism's cells are getting replicated to replace old cells, for example. And then living things today also have structures. They're not uniform homogeneous substances, to use maybe a outdated term. They have organs in the case of multicellular organisms or multicellular animals in particular, but even unicellular organisms have organelles and membranes and different structures. And maybe this is not an essential feature of life in thinking very expansively, but certainly it's a big part of life here on Earth. And so the challenge then is to figure out how can a physical non-living system or molecule or aggregate of molecules perform some of these functions, or at least one of these functions. And if we could do that, then we could perhaps identify something that could give rise to life either here or elsewhere in the galaxy. And of course, if we find such a physical system elsewhere in the galaxy, then we might be onto something. We might find we might find life or something that could become life someday. So it's very interesting if we can do it. And there are two candidate molecules that might perform one or more of these functions. One is RNA, the molecule on the right here, very creatively imagined, emerging from this lovely green soup. And this is the one of the older hypotheses about how life might have emerged. It's over 50 years old. And the thought is that you had a kind of friendly liquid environment in which RNA molecules formed, and RNA molecules are really nice in that they can, it's reasonably straightforward to figure out how they could replicate. And metabolism and compartmentalization are functions that we could perhaps better understand if we felt sure that RNA were indeed the precursors to life. But it's a bit of a difficult molecule because it's not a very stable molecule, unlike DNA, and it doesn't necessarily form very easily. So getting the conditions just right for RNA to form and to stay stable long enough to replicate and to evolve into life seems like a bit of a challenge. And it's worth considering alternatives. And a relatively recent alternative, 20 odd years old, is what's called the lipid world hypothesis. And the idea here is that it's not RNA that gave rise to life, it's actually lipids that first emerged and then later evolved into living organisms. And the nice thing about lipids is that they're quite stable, and they form these structures. As you can see here, these are sort of cellular membranes. They form these quite easily and stay stable. So you've got compartmentalization, which was one of the important functions. And if we could establish that lipids could replicate and metabolize, then we've got three functions that life today perform in one type of molecule, and that would be a very promising indeed way to make headway with the origin of life. So the challenges are to better understand this hypothesis. Can we establish that lipids self-replicate? Can we establish that they compartmentalize? And can we establish that they metabolize? And the compartmentalization is reasonably straightforward in the sense that we've figured out, we have laboratory work showing that it's pretty easy to get lipids to do this, to form structures that are compartmentalized in the right way. So how do we establish, though, the replication and the metabolism? In particular, replication seems like a really central feature of life, and it's hard to get lipids to do that in the lab, or at least we haven't been able to, as far as I'm aware. And so we turn to modeling. Modeling is a fruitful way, potentially, of trying to understand something that we can't do in more physical terms. And the GARD model, the graded autocatalytic replication domain, we'll just call it the GARD model, it hopefully makes headway with that. And as you can see, it's a bit of a complicated model, and I don't want to get into the details just now too heavily, but I'm happy to talk more about it in the discussion period. But I think we can understand some of the important features of this model, and helpfully there's this nice little diagram that the modelers have developed that I think is quite helpful. So the idea here is that you've got a liquid solution in which there are a bunch of small lipid molecules, the lipid monomers on this slide. And some of these lipid monomers will spontaneously join together and form an assembly, as it's called. And this is just a cluster of lipids. And it's a fairly stable structure. It does lose some lipid monomers over time. That's one of the K terms towards the top. But then some of the lipid monomers from the environment will also just latch on from time to time. So there's some small change over time in the composition of this assembly. But crucially, some of the components of the assembly might actively facilitate the acquisition of new molecules. And that's what this beta term is here. And what exactly beta is depends upon what the assembly is made of. But the general idea here is that you've got an assembly and it's able to actively acquire a material from the environment and to grow and to eventually split apart and to form a new assembly. And that's interesting for a number of reasons. And I can talk more about the mathy stuff if we're interested, and the technical details. And there are also, it's worth pointing out, slight different versions of this model, and it can be used in a variety of different ways. But I'm going to set that aside for now, and we can return to it if we would like to. I'd like to point out that there's a key assumption that this model has, and that is that there is a mild environment, a liquid environment, and that it is rich with lipid molecules. And I'm going to come back to this key assumption because I think it's quite important. So with this model, if you run it through a Monte Carlo simulation, you can show a number of interesting things, as Lancet et al. Have done and others have done as well. And that is that if you've got the right starting conditions, that is if you've got a mild environment with lots of lipid molecules, you can get them to form, provided they're the right kind of lipids, form into these structures. And furthermore, that structure is autocatalytic. That is, it self-sustains. That is that beta term. It can acquire new material from the environment and stay stable. And lastly, and most interestingly, I think, is that while it's in that steady state, these lipid assemblies can reproduce and pass on stable information. And if you've got that, then you've got the potential to have evolution. Now, the information stored here is a little bit different from the information that we know today in DNA, for example. So DNA stores information in base pairs, different variations in base pairs. The assembly here stores information in ratios of lipid types. So I'll just return to this slide here for a second. You can see that there are lots of different colored balls here representing the lipid monomers. So each one is a different type, and the ratio of those different types is a kind of information that is stored in them. And so if you've got the right ones, and if you've got the right environment, that ratio will stay fairly consistent, even through splitting into multiple assemblies once it's big enough. Now, that's of course quite different from life today, but we don't need to show how precisely at this early stage you get a lipid structure into life. We just need to establish how can it perform all these functions, and then future research will establish those other things. However, there's a sense in which this model has no target. That is, the model does not describe anything that is in the actual world. So philosophers have traditionally thought of a model as a man-made object of some kind, and that thing that they've made, that model, has a representational relationship to something in the world, some sort of physical system or object. And what makes models interesting is that they've got this representational relationship. You've got the model and you've got something in the world, and there's a connection between them. However, for this particular model, there are no lipids that have the properties that the model describes. So I don't think we can say that the model represents any actual assemblies or lipid monomers. And of course, it does not also represent the origin of life or the precursor to life, because we don't know whether life did in fact emerge from lipids. And I think this poses a challenge, which is, how are we to understand the value of this model if it doesn't allow us to reason directly about things in the actual world? And I think because we don't know whether it describes things in the actual world, it's useful to come up with a classification for this kind of model. And I'm going to call it an indeterminate model. Okay, and a model is indeterminate if and only if, firstly, the model is without a clear target, which I think this one is, right? We don't know whether there are lipids that meet its criteria. Secondly, we're not in a position to rule out the possibility that it might have a target. So it could be that later down the road, we discover that life did emerge from lipids. We might find this on another planet. We might recreate it in the lab. There might be something else we're able to do, but it's possible that life did emerge from lipids, right? So we can't rule out that possibility. And then thirdly, the model doesn't allow for modal or analogical reasoning. And I'll say more about this towards the end of the talk, because I think that situates this model relative to some of the other things people have said about modeling. So why is this important? I think this is important because the model doesn't, although it doesn't allow us to reason directly about the natural world, what it does do, it allows us to constrain uncertainty, and it does that in three different ways. And what I'll do is I'm just going to outline those three different ways, and then I'm going to illustrate how it has done so in the context of astrobiology. So one thing the model does is I think it allows us to identify sources of evidence. So it points us towards what we need to be thinking about, what kinds of missions we need to run, what kind of experiments we need to conduct. Where do we find the evidence to make headway with this question of whether life emerged from lipids, or could emerge from lipids here or elsewhere. Secondly, it offers standards for judging and interpreting new evidence that we might find, but then also old evidence that maybe we didn't realize was significant in certain ways. And then finally, and perhaps most influentially in the literature, the scientific literature, the model provides justification for the legitimacy of the research strategy. And I think all these things are made possible by having uncertainty over whether the model has a target. I think if the model did have a target, or if we knew whether it had a target definitively, we wouldn't be able to do these things. And I'll say a little bit more about that as well. Okay, so how has the GARD model done these things? Well, I think it has done so in the context of this very interesting astrobiology mission. It wasn't just astrobiology, but it has astrobiological significance. And that mission is the Cassini-Huygens mission, which did a flyby of Saturn's moon Enceladus. It did a few flybys, and in addition to taking these beautiful photos, such as this one in 2015, it also collected a lot of data about the planet. I can talk more about that later, but it collected tons of information, and scientists back on Earth were able to analyze it and do a number of interesting things, one of which Postberg et al. Did in 2018 was to show that Enceladus has a lot of organic molecules in a probably liquid or probably water liquid solution underneath an icy crust, which is interesting for a number of different reasons. It's inherently interesting just that there would be liquid water so far from the sun, and that there would be organic molecules in that liquid. But crucially, I think, for this question of the origin of life and how life might emerge, it seems to be an example of a kind of primordial soup. Not that there's been any direct evidence of life or anything like that, but it seems to be a relatively mild environment in which there are a lot of organic molecules. And Kahana et al. Argue that this actually is really very important for our understanding of how life might emerge, because it shows the possibility of a primordial soup emerging in unlikely conditions. And that, you may recall, is one of the key assumptions of the GARD model, that you have a mild liquid solution in which there are a number of organic molecules, especially lipid molecules. So one of the assumptions of the GARD model seems not certain, of course, right? Because Earth might not have had a primordial soup, but primordial soups don't seem to be an outrageously implausible thing to have emerge. And the significance of this finding is made relevant to origin of life studies, in part because of the GARD model. So I'm just going to return to those three ways in which I thought the GARD model allows scientists to constrain uncertainty and structure research. Okay, so the identification of evidence. So the GARD model offers an avenue for constraining uncertainty by identifying what further evidence is needed and where to look for it. So because the model had as a key assumption a kind of primordial soup, it then points us towards the significance of discovering something like a primordial soup elsewhere. It tells us primordial soups are going to be potentially very important for understanding how life emerged, and so we should be looking for them and studying them. And in particular, perhaps looking for primordial soups that have a lot of lipids instead of say other kinds of carbon-based molecules. So it identifies useful forms of evidence for us to pursue. That was one thing. A second thing is that it allows us to provide to interpret evidence that we find in a particular light. And I think that's what happened with this Cassini-Huygens mission. The GARD model gave us some resources for thinking that, for evaluating what the mission was bringing back to us and showing that it was significant for how primordial soups might emerge in different kinds of systems. And then finally, and I think this is perhaps the most influential thing that it has done, is the GARD model gives an affirmative answer to the question, is this line of research worth pursuing? Is it worth trying to establish whether life could emerge from lipids? And that, of course, is an important question to ask. There are lots of things life might have emerged from, we think. Is it worth pursuing this one particular line specifically? And I think the GARD model suggests that it is worth pursuing that, because it seems like lipids in principle could perform those three functions that we find in contemporary life: compartmentalization, replication, and metabolism. So it shows that that line of research is plausible in a way that might not have seemed plausible before. And I think it does all these things without having a target and without allowing us to say anything definitive about actual lipids, right? Because we haven't found actual lipids that form assemblies in this way. So just to recap this, the GARD model allows scientists to constrain uncertainty, that is, in facing an uncertain path forward to studying the origin of life and to searching for life elsewhere. It allows us to provide some structure to that uncertainty by identifying where we should look for evidence, what kind of evidence we should be looking for. It gives us resources for interpreting that evidence, and it provides justification for thinking that that's a useful way of going forward. And I think all this is made possible by the fact that we don't know whether it has a target. And this is shown, I think, by contemplating what would be the case if it did have a target or if it did not have a target. If we knew the GARD model did have a target, that is, if we knew that lipids were the precursor to life, then a lot of this, the model itself wouldn't necessarily be very interesting, right? Because we'd be trying to figure out what specific lipids they were. We'd be trying to recreate it in the lab. There would be lots of other things we'd be trying to do. And we wouldn't need to be looking in the same way. We wouldn't be needing to look for the same things that we're currently looking for. On the other hand, if we knew that life did not emerge from lipids, then of course this whole project then is not worthwhile, right? Because we would know that it's not the case that lipids form life. So really, we need to this model to not, we need to, in order to find this model interesting and valuable, we need to not be sure whether in fact it does represent, that is, whether it has a target. We need to not be sure whether there are lipids that can form assemblies that replicate in the relevant way. And I'd like to contrast this view, I think, with a few other ways we might think of, two in particular ways we might think of this model as providing valuable knowledge. So one influential way of thinking about how a model without a target could provide knowledge is through a how possibly explanation. And this is a pretty old debate, but there's been some really interesting recent-ish work from Bokulich and Forber looking at how possibly explanations and modeling. And the idea here is that for some kinds of models, they don't necessarily tell us how things actually are, but they tell us something about how it might be that a phenomenon came about, say, or how it might be, how possibly a cause works, or what kind of causes are at play. And this is, of course, very interesting, right? Because if we knew what was possible and what was not possible, then we're able to hone our research in, and we're able to generate some predictions that could be testable. And Bokulich in particular, I think, has done some excellent work with this. She has a very nice case study looking at the tiger bush, which is, there's a photo of it on the right here. The tiger bush forms these growth patterns that look a bit like tiger stripes when viewed from the air. You can see it a little bit here in the photo. It's sometimes more and less or less dramatic. And it's very mysterious why they form these stripes. And there are a number of different models attempting to predict it. And some of them are more successful than others. Some of them are more abstract than others. And these different models, this sort of family of models that are representing the tiger bush, are able to help scientists by showing what possible explanations there are. So, right, if a model doesn't give this kind of pattern, then, well, we know that model is not good. If there are 6 models that do, well, maybe one of those 6 models is right, and so we should investigate those further. So it tells us they tell us how possibly this phenomenon could have emerged. And I think this initially seems to fit well with the GARD model, right? Because you might want to say, well, the GARD model tells us how possibly life might have emerged by establishing that lipids could perform these 3 functions. However, I think that that's not quite right, because it's a few steps ahead, right? We don't know whether the GARD model does tell us how possibly life have emerged. We don't know whether it is possible life has emerged from lipids. If we found lipids that have the properties the GARD model ascribes to them, then it might be the case. Then maybe it would be a how possibly explanation. But I don't think we've gotten there yet. So I think these kinds of explanations work quite well. That is, the how possibly explanations work quite well for models with a much higher level of certainty associated with them. The other, and I think it's closely related, kind of treatment you might give the GARD model is one associated with modal reasoning. So Massimi argues that for models that don't have targets, they're valuable because although they don't have a target directly, we can use them to ascribe modal properties to actual real-world phenomena, real-world physical systems and objects, particles. And she's writing about the hunt for new particles at the Large Hadron Collider at CERN. And she writes, exploratory models ultimately deliver important modal knowledge about what is causally possible concerning a phenomenon under study by attributing to a fictional entity some essential properties and relations, which, by a suitably keying, are translated into essential properties and relations imputed to the phenomenon. So I think we can just sum up this general position here by saying a model gives modal knowledge if and only if scientists use it to ascribe modal properties to actual phenomena, possibly via a fictional system. And I think this also initially seems to fit well with the GARD model in the sense that it seems like These lipids, the model describes, are fictional, so they're part of a fictional system. That is a system that's doesn't actually exist. And yet it seems like at the same time, scientists might be able to use that fictional system to ascribe modal properties to, say, the origin of life, say. And the modal property might be that life might have emerged from lipids. However, I think this is also a few steps ahead of what the GARD model is doing, and that's because there are no properties, modal or otherwise, ascribed to actual phenomena using the GARD model. That is, we don't yet know whether it is possible life emerged here from lipids, and we haven't found evidence of that elsewhere in the solar system. So I think these views capture quite nicely what's going on in cases where there's much more certainty, or there's much less epistemic uncertainty, at least, about what it is we're studying and also how it is we should study it. And I'd just like to propose here that thinking about indeterminate models like the GARD model and some of the other treatments in the literature of models, it might suggest that we could classify models based on the degree of uncertainty that we have about the research. And there might be a spectrum. On one end of the spectrum where there's lots of uncertainty, you might have indeterminate models like the GARD model. And in such a case, you learn something directly about the actual world, but what you do is you structure your research for looking for new evidence. You have resources available for interpreting evidence as it comes along, and you've also got some justification that you're on the right track, that you're doing something interesting that might yield results down the road. And then if you've got a bit more knowledge about what you're studying and how you're studying it, then your exploratory or fictional model, it might give you modal knowledge of what you're studying. So you might not know, in the tiger bush case, you might not know exactly how these patterns emerged, but you might know the modal space. You're right. You might know what the options are, and you might know how it could not have formed. You might be able to rule out some possible lines of research. And then if you've got tons of knowledge, you might be able to just ascribe actual properties to the phenomena. I think a ball and stick model of a water molecule might be a good example of this, but there are tons, right? But the ball and stick model allows us to reason about the number of bonds that atoms can form, and also about the angles of the bonds, for example. And those aren't just modal properties; those are actual properties of atoms. We think. So, if that's the case, then I think there's a lot of nuance that we might find in different kinds of models that might superficially seem quite similar based on, for example, whether they have a target and whether they don't have a target. And those are the main things I'd like to cover today. And I'll just recap what I've hopefully accomplished. And one is that the GARD model is indeterminate, which means that we don't know whether it has a target or not. And that indeterminacy is really helpful because it allows us to constrain uncertainty, and it constrains uncertainty by pointing us towards the kinds of evidence we should be acquiring. It gives us resources for interpreting that evidence, and it provides justification for that particular line of research. And I also think that it gives us reason for thinking that there's a lot of nuance to different kinds of modeling practices that seem quite similar, and that it might be uncertainty is the criterion for distinguishing different kinds of models. Here are just some image credits and some selected references. Thank you very much for coming today, even in the face of the strike. I'm really grateful to have had this opportunity, and I'm really looking forward to hearing your questions and thoughts on this. Thanks very much.